Good. Welcome back, students, to the class. We'll be discussing lecture 5, Introduction to Genome Sequencing. So I divided the class into an outline. You see the points here. Briefly, we will talk about the genome size and genome organization. Most of the points are very briefly we will talk because it's a general introduction to genome sequencing again. But it is very fundamental to better understand the whole Unit 2 that talks about a lot of stuff about human genome projects, express sequence tags, RFLPs, SNPs. A lot of technical jargon will come up. Unless you don't understand the fundamentals of how sequencing is done, it is very tough to follow up in further classes. So I divided the outline into following topics. We will study today genome size and organization, followed by just brush up our basics of RDNA technology so that we know everyone is on the same page. Then we talk about DNA labeling briefly on polymerase activity and methods of sequencing that developed over time. I think Sanger sequence is one of the best known method that was identified in 1970s. From there, the sequencing methodology in terms of evolution of technologies, along with that, a lot of bioinformatic tools were added. So we are going into first, second, third, and presently a fourth generation of sequencing technologies are available. We'll briefly talk about how the trend happened and what are the advantages in terms of the cost and functional significance of the genome sequencing technology. That will be the essence of the class today. Let's take an example of human genome. How it is organized, what is the size. You just briefly look here. So we know that the total length of human genome is over 3 billion base pairs. See that it's not small, it's very huge and long, 3 billion base pairs. And the genome is organized into 22 paired chromosomes. We are diploid, so we will have one copy of each of the gene in terms of chromosomes. 22 paired chromosomes called autosomes and one X chromosome, depending on the sex of the human being, XX and XY. You see that here. And these DNA molecules are usually compacted into chromosomes, but these are all large linear DNA molecules contained within the position of the cell is called nucleus. So that is that summary of 3 billion base pairs or 3,234 mega base pairs, million mega base pairs, that you can define the length of the human genome. So compared to in evolutionary scales, where do we stand up with other species and other genomes? You look in this table, the organism here, genome size here, and the functional genes estimated to be coming out of the genome of an individual species. For example, human, we are here 3 billion, 30,000 estimated genes. And take an example of roundworm or C. elegans, the genome is 97 million base pairs, and then bases or base pairs. It's bases. We're not talking about base pairs here, single genome bases, and 19,000 estimated genes. And what this table tells us that though the genome size varies, it is not corresponding or proportional to the estimated number of genes. So though we are at 3 billion genome size, but we have 30,000 estimated genes, but the C. elegans roundworm, phylogenetically a distant animal from humans, it has also close to 19,000, not much difference here, but there is a huge difference in terms of the genome size. So basically the genome size doesn't correspond with the coding or estimated genes coming from the genome, but it varies across the phylogeny. And also this is not an hierarchy. It's not like we are going from simple organisms or simple genome size and going into complex. It's not like that. If you see one of the protist, you can see it's also has billions of some close to 1 billion in size for the genome. The size is also doesn't proportional to the phylogeny, and the estimated genes are also. It's just giving a simple example of the genome organization. At the same time, we know that the nucleus, the DNA that consists or that hosts the DNA, the blueprint of any human being, any species. And you see that there is a very compact organization of the genome into the DNA, into the nucleus, for example, in a cell. And we already know that when you zoom in, you go from DNA, the whole, it's kind of a spaghetti, right? It's clustered into a spaghetti, ball of iron. And then you take into different chromosomes, it's organized. Then there will be a complexity in terms of structural complexity, in terms of solenoid loops. Then the loops will go and tie up. When you zoom in, you're zooming in from chromosomes to loops and solenoid loops to, again, you know that there is a nucleosome organization. Histone proteins will help in the packing of the DNA. And you zoom in again, you go into the individual gene. Let's say an example, you stretch the DNA, probably the whole cells of the body and the whole DNA coming from all the cells. You stretch it out into a single thread, it would come into the between a distance between Earth and Moon almost 6,000 times. So you imagine that a human body, just such a long macromolecule, for example, the nucleic acid here, that being condensed into a small nucleus that's probably a location of 100 micrometer or distance, something the length of the cell here and the nucleus. So basically, in talking in literally sense, DNA is short, at the same time it is long. Human DNA is small and large at the same time in terms of literally speaking. And this tendency, this kind of condensation also makes us difficult to access the DNA at the same time. Let's say you want to see a stretch of a gene, you want to identify the nucleotides, you need to have a technology that can break the membranes, that can take the DNA in isolation and also in a pure form. That we already know from molecular biology techniques. We just go into basics of RDNA and molecular biology a little bit in the next few slides. We already know that the DNA is a double helix. The double helix is coded by specific nucleotides called adenine, thymine, guanine, and cytosine. They will have a base pairing or complementarity with each strand, and then they are bound by specific phosphate bonds. We will see in the next few slides. This is the basic organization, fundamental units of the DNA, so nucleotides. You can read through the slides and read through the text to brush up your fundamentals. But remember that there is a base pairing. The base pairing and complementarity is one of the fundamental aspects where we took advantage of this base pairing and started from this only to went into different technologies and different sequencing methods. Let's zoom into one of the functional unit of the DNA, nucleic acid. It's called deoxyribonucleic acid, right? DNA. So it's made up of basic units called DNTPs. DNTPs are deoxynucleotide triphosphates. Deoxy. So you start with the pentose sugar, that is the backbone of either DNA or RNA. So you see here, there is a kind of 3' OH, 2' OH, and one of the pentose, one of the 5 branches. The bonding is there. 3' free hydroxyl, 2' free hydroxyl is there. These 3 and 2 both are hydroxyl groups in terms of the ribose nucleic acid molecule. But the pentose in the deoxyribonucleic acid it's a kind of a deoxylated. So you get only H here in the 2'. You get a free hydroxyl group at the 3' OH for deoxyribose nucleotide triphosphates. And you say that a pentose sugar, just without the phosphate bonds, 3 phosphate phosphodiester bond will be there and 3 phosphates will be here. Without that is called nucleoside. The pentose sugar with 3' OH and a 1 phosphate group is called nucleotide, and then diphosphate and triphosphate. So nucleoside triphosphate. So nucleotide triphosphate, we call it as the deoxyribonucleotide. And then the base here changes with the purines and pyrimidines. Purines are adenine and guanine. Pyrimidines are cytosine, uracil, and thymine. Okay, it's clear. This is you should be able to understand it better to further go through the slides with a small pace and with better understanding. And also we know that there is a DNA polymerase enzyme. That enzyme will kind of helps in extending the DNA along with the 5', 3' direction. Let's look in 2 strands here. This is a template strand in the blue. It's running 3' to 5' direction. And then RNA reads through the template strand to 5', 3' direction. The enzyme is able to catalyze or add additional nucleotides to elongate the DNA along here. This is a new strand generated. That's a template strand. And we should be able to remember that it cannot start de novo. It needs a priming process. It needs a DNA primer that has been already established in the system. From the primer, it extends the template strand. And in the free soluble solution, we can give DNTPs. It adds the DNTPs complementary to that. It is a fundamental concept in that. You should remember that how this extension happens. We talked about the 3' hydroxyl group here. It attaches to the next nucleotide molecule that is bringing a one more phosphate in the first position, there will be a phosphodiester linkage that makes the DNA attached to the one nucleotide to the next nucleotide and elongate further, and these two phosphates will be released in that. So this is the direction synthesis happens, and it was observed that when you take this 3' OH group out, for example, you make a DNA pentose that will have both 3' 2' dideoxyribose, where there is no oxygen molecule present here. We came to know that this reaction of extension of the DNA in the new strand in the direction stops because there is no free hydroxyl group that cannot bond with this phosphate. You get 3' 2' dideoxynucleotide that will not extend the synthesis process. It basically stops the synthesis wherever this form of hydrogen present in the pentose. So this is called dideoxynucleotide molecule. This is deoxynucleotide molecules. Please try to understand, make notes for this. This is one of the important steps. You can write in any of these 2 mark, 4 mark and other essays given in the exam. You need to start explaining on this and then extend your understanding to the specific sequencing method. So dideoxynucleotide and stopping and a reaction of the nucleotide extension was first discovered by Dr. Fred Sanger. He's a double Nobel laureate. He's the one pioneer developed this dideoxy sequencing method, first published in 1977. And so we should appreciate the efforts of this an individual person, individual perseverance, motivation, dedication, try to understand a specific biological process. So he went beyond expectations, just as like described by one of the person here, Bradley. Fred Sanger is a quite giant whose discoveries and invention transformed our research method. Absolutely, they transformed the whole genome analysis, genome sequencing method from 1977 over 4 decades of timeline. So again, one more process you need to understand that. So deoxyribonucleotides can be attached to a fluorescent tag. So for example, the fluorescent tag usually comes along in this structure. The base comes here. The tag can be attached to one of these. You make a redox reaction, you make a reduction. One of the bonds will be exposed in pyrene, puribulins. You attach a specific fluorescent tag to that. Then either the tag may be green, red, or whatever based on the fluorescence molecule or probe available. You attach the probe to the base, then you can recognize the base irrespective of the 3' hydroxyl, or you stop the hydroxyl group, then which base it stopped, you can easily recognize based on the probe or fluorophore you attach it to the base. And alternately, people sometimes try to attach to the phosphate group, try to take advantage of this elimination of the 2 phosphate groups out of a reaction when extension happens. So they took advantage of that for understanding some other process of synthesis. But you remember that the base can be fluorescently tagged. When it stops with the hydroxyl group here in the 2', 3' dideoxy molecule attaches, you can recognize the base based on the fluorophore signal. And the basic of recognition that goes through energy, ground state to excited state, energy comes up, then shorter wavelength, relaxation, the fluorophore and the tagged molecule can go into that de-excitation. This change in wavelength can be captured by a light that gives a specific fluorescent signal corresponding to the tag or wavelength of the tag we attach it to. And also coming back to fundamentals of polymerase chain reaction, Mullis was the one discovered polymerase chain reaction. It's a very simple concept, but it took some time to just understand the process and make it into a successful event. But it's a simplistic way you understand that. You have a copy of DNA in vitro, for example, in a tube, you have a DNA isolated. You have a specific length of DNA that is put it into the test tube. You want to amplify the DNA. We know that now DNTPs are there. There is a polymerase enzyme you can amplify. So you go through specific steps of polymerase reaction: denaturation, annealing, and elongation. Denaturing, primer annealing, elongation with the polymerase. At the end of the step, you exponentially start with the same set of DNA that in the tube. You make multiple copies of the same DNA through a PCR reaction. You generate huge amount of the DNA. The fundamental question is, when you are adding it one tube of the DNA, one tube of the PCR machine or PCR tube, you make several copies of the same template DNA you added into your tube 1 in each tube. Like that, a PCR machine will have 100 wells, 100 it can locate or it can accommodate 100 tubes. You can add 100 copies of different DNAs into 100 tubes. But remember that each tube will have only one template strand, one copy of the template strand, and it keeps making multiple copies of the same. It doesn't change it into a different form. All the nucleotides are of the same, but the volume increases by exponentially with the PCR machine. This is also an important step you need to understand. So again, going back into a family tree of sequencing methods, molecular methods, we know that we know that cloning we can do, and we know that the bacterial viral vectors are available. You can use a vector for cloning. You can put an artificial insert of gene, either antibiotic gene, or insert your PCR DNA by ligation, restriction, digestion, and cloning. At the same time, that's one more fundamental of the tree or any methodology is DNA is the first thing is that hybridization of DNA, homodimer, heterodimer. Homodimer is DNA to DNA, hybridization into heterodimer, DNA to RNA, and the development of the probes. That is one of the fundamental foundation that made us to understand better on the PCR technology, other that evolved to qPCR, and methods to analyze agarose gels. You go with the cloning, and at the dead end, at the end of the chain, we are looking at a sequencing methodology. Try to look into that. Different again hybridization methodology, localization of the DNA through microscopy, qPCR quantitate, and also study DNA RNA through sequencing or footprinting agarose gels. You can do that. That's the way we evolved, and we are presently in the sequencing platform where you can stop the primitive processes here and then directly go into an advanced method. So by going so, you can go out of these very hardworking steps, right? For example, to clone a single gene in 1980, it's not an easy job. It takes some years and years of time and a lot of effort from an individual or a group of graduate students, undergraduates, scientists working together for to clone one gene. It may have taken 1 decade of time. But with the technological automated sequencing, we will talk. You can escape all these steps, which are laborious. You just go into a simple, just give your sample, get the data out in a day or in an hour. We are evolved into that way, but making laborious process into convenient way for understanding and going for genome sequencing. So just a glance, we will go in next 10-15 minutes, understanding advances in genome sequencing methodologies. So this is a stretch of DNA sequence that automatically came from a machine, not running through a PCR gel, running through PCR, running through agarose gel, again doing it, the staining, and again understanding the size and which are the bands. You don't need to do that. You just simply given a sample, put an automated sequencer, then you found out that the DNA sequencing can be given a stretch of 180 to 530 nucleotides, probably 300 nucleotides here. And the machine just tells you that what is the nucleotide position in this, what is the position at the 531. And it just gives a specific peak based on the fluorescence signal, and it tells you that exact identity of the nucleotide in the template strand. Remember that this is a complementary coming. The sequence is complementary. This is a template strand. So how it can be done? How this simplistic way of analyzing DNA sequencing was done? How it was came into in terms of what methods we used? As discussed previously, that you can attach a fluorophore. For example, I am talking about a nucleotide here, a sugar base, pyrene or pyrimidine here. You can attach a green, mainly guanine is attached with a green fluorophore, adenine with yellow, and then orange for cytosine, adenine for thymine for red. So you attach a specific fluorophore, you can recognize based on the signal. So one of the methods that used for this was Sanger sequencing, 1980s. This is an example of automated, but we will talk briefly in the general how the papers published in 1970s look. But basically try to understand the method. So this is a stretch of template DNA given to you. And at the end, you are looking for what is this sequence coming from here, because the template DNA, you might be given as a crude DNA sample, right? You cannot visually see the nucleotides. You are isolating from the cell. You got a DNA sample, you measure the quality of the DNA, but you don't know absolutely what are the sequences starting from one length to one stretch to the other stretch of the DNA. So unknown sequences in a given template DNA can be seen in the chromatogram. This is the output chromatogram based on the color. You read the chromatogram visually, you can tell which sequences are where they are positioned. How you can do that? Basically, by doing the hybridization, doing the extension of the template DNA. Basically, you know that there are 4 nucleotides only that extend, that constitute the whole DNA. So you give them with different color fluorescence tags. You give them in vitro. You make artificially these synthetic molecules. You give them in a test tube. And then at the same time, as Sanger explained, you can stop the synthesis reaction anytime. You can give again dideoxynucleotides also. You can also label them. You can give them in a solution. But you remember that you try to that these DDNTPs go and stop this reaction at a random steps. They doesn't stop at, for example, this is a 13 nucleotide template DNA here. So it is very randomly goes and stop the reaction. Remember that you have a template, same template DNA from the beginning of any reaction in vitro. One tube will have same DNA. When this DDNTPs stop this reaction at varied length, for example, they may stop the reaction by attaching to the thymine here. So if they stop from, because you are going from this direction to this direction, they stop at the T here, you have a 1, 2, 3, 4 DNA of 4 nucleotide only. That may be here. That may be here, talking only T1, a combination of 1 plus 3, only T here. So you may get a only extension of 4 nucleotide only, given a sample of the same length. At the same time, you are exaggerating or you are bombarding the machine or in vitro culture with a lot of excess of this DNTP. So by allowing that, they may get multiple varied length of the same template, a combination of 4, 6, 8, 10, but the longest combination you can get is including this to that 13 nucleotide length only, based on the length of the template or based on whatever hybridization can happen, right? It cannot exceed more than that because the template doesn't have more nucleotides here. You get a varied combination of this extension of the length nucleotides coming through this hybridization reaction. Then you can separate these nucleotides by length in agarose gel electrophoresis. The smallest molecules of DNA goes first and they get detected to a laser beam multiplier, and largest fragment can be on the top of the gel based on the size. Then you say that, you run through that, you know that that is present here is a G, A, C, T, T, T is the present in the bottom here. But remember that this G, the DTTP stopped here. The G stopped here, that has a stretch of 13 nucleotides. It is heavy compared to the G present here, compared to the T present here. This T is heavy. For example, it has longer reads. And then by doing so, you generate clonal population, or you generate clones of multiple varied length of the same template DNA. And then you will have, for example, you may get 1,000 copies of the same template with different lengths like this, 1,000 or 500 combinations. Then the software can detect it. It can go into a stitching process, or consensus can be established. At the end, you can, going through all the copies multiple times, can say that yes, it is a 13 base nucleotide length, and first nucleotide is G, last nucleotide is T. That is the fundamental concept of Sanger sequencing. It's an automated way. It can go for 400 reads of DNA per sample, and then given a capacity of the machine, you can sequence 1 million sequences per day in this Sanger sequencing automated machine. But remember that 400 reads of DNA per sample. So we are looking 13 stretch DNA here, but the method Sanger can sequence up to 400 reads of a single stretch of DNA given per sample. And then you can put multiple samples per day. The machine can sequence 1 million sequences. So again, this is the agarose gel, was done in 1970s. You can see the length from larger fragment to smaller fragment, location of the AGCTs. You can run them based on this fluorescence tag separately, GATC, and then you combine them into a single gel, right? You take that, this is the first position, you go into the position later here, you add 1, 2, 3, 4, 5, 6, the gel becomes 1. This is the way traditionally done. The sequencing output comes here. But nowadays they can put it into only one gel only through capillary electrophoresis. You can put a combination of that. It can, based on that, we know only one well, it can generate, it can identify the stretches, and it gives a chromatograph based on that. This is another graphical representation of the same method. You can go through again, understand in a better way. So again, quickly going through the historical perspective of molecular biology, tree of methods, a family tree of molecular biology methods, how the trends changed and what made into present situation of next generation DNA sequencing. In 1950s, first time we were able to understand electrophoresis, agarose gel or protein SDS page, agarose gel. 1953, double helix was identified. Same way you go into better understanding, there is a mutagenesis, there is a DNA fingerprinting, PCR method was invented here. Sanger was invented in 1975. Then we can into 2005, next generation DNA sequencing, the most highly advanced method came up here. And our genome was sequenced in 2001, draft of the human genome was sequenced here. So as methods evolve, there is a kind of effort from scientists all over technological revolution. We go into simplistic way of understanding the genome sequence. For example, Human Genome Project, it took 20 years at that time. It completed in 2001, but there is a consortium that developed a decade or 2 decades over. It took 20 years. It took close to $3 billion US money, and it came in 2001. And post-genomic era, after this human genome, from this 3 billion, the cost went into 100 million, and because of the automated sequencing method. Just a brief overview, look into that, appreciate the efforts of various scientists to make a simple next generation DNA sequencing methods. Remember that we know that Sanger sequencing automated also 400 reads per, it goes into 400 reads per DNA sample, 1 million sequences per day only, right? And also remember that human genome is 3.2 billion. And also if you go into manually or automated way, so remember that how much time it can take. If you take one machine automated Sanger sequencing, go with this sample limitation, you may take 100 years to sequence the human genome. So to avoid these complications, what they have done, they invested huge amount of money into that. They made factories of the DNA machines, automated Sanger sequencing, so that multiple parallel copies of the DNA can be sequenced. And then you see the well plates here. You may have a code for the machine, code for the plate, automate the data. So simply warehouse. A lot of money was invested to reduce the time and efforts. And then parallel machines have run in 24/7. At the end, we were able to sequence the human genome. So this slide talks about approaching to next generation sequencing. Again, it talks about the evolutionary trends. Just briefly go into the which year, what happened. And in terms of they're talking about evolution and the method of DNA sequencing, 1977 Sanger sequencing was there. And 1993 another method called pyro sequencing was developed. You along go with that, there is another technological revolution called cluster DNA, multiplex PCR, emulsion PCR that we may briefly discuss if required for the curriculum. So remember one more time, the Human Genome Project provided an impetus for technological innovation in DNA sequencing that led to next generation sequencing methods. So, and also remember that the human genome was where the sample came from for sequencing human genome. The consortium has taken a few individuals only. They collected DNA from few individuals. They went into effort of sequencing them. That took 20 years and huge amount of money. Then what about the other, what about all of us? Why we need to prefer some other individual genome? Why not my genome? So that was the question that came, because it's functionally understanding the genome is really important for personalized medicine and many other things that we will talk in next class. So they made that they want to make a simplistic way of sequencing the genome. So they went into these evolutionary trends. And you see the cost, how it was reduced, we will see in the next few slides. So I have less than 5 minutes. I will try to go through as quickly as possible now. You read through the slides in your free time again. So basically, sequencing technology, DNA sequencing, classical approach, Sanger reaction, next generation sequencing, massive parallel sequencing or high throughput sequencing. You have different molecules, different methodologies that evolved through that. Output varies. A single sequence ranging from 500 to 1000 base pairs from Sanger, you may go through output it kind of gigabases of sequence ranging from 25 to 500. You are going with the gigabases, producing an enormous amount of sequences, high throughput. Again, read through that, different methods of NGS sequencing here. Go through that, how the samples are prepared. Go through this step: amplification, sequencing, and imaging. Sequencing either pyro, sequencing by ligation, sequencing by other terminators. We might see some of these fundamentals required, but get an overall understanding of this. Again, which sample of the DNA you can use? What is the source you want? Genomic DNA, gDNA, total RNA, targeted DNA sequences that you can give them to a machine in the initial process. And then you can go for sequencing methods that may be either single read, paired read, or multiplexing sequencing. Single read is that you know that the DNA is a template, double standard. You give the primer, you read one sequence only from one end to the other. You don't do with the reverse end, that is single end reading, and you will have a database. And then paired end reading, same DNA, you go in one direction, again you go back in the other direction. You sequence in both strands so that you see the complementarity exists or not. Multiplexing is again, it's a pool of DNA. It's not directional. It's directional, but again, not a single stretch. A lot of pooled DNA can be given, and a lot of pools of DNA can be simultaneously sequenced, not a single stretch of DNA or double stretch of DNA. A varied pool of DNA can be given. All pools can be sequenced at the same time. That is multiplex sequence. This is a method for multiplex sequencing. You will have a kind of what is this called, binders or DNA fragments with the binders and the template DNA. The binders will have a barcodes. You see that now you are moving into barcode technology. And these binders, one binder, second binder, goes and randomly binds the genome sequences coming from gDNA. And then this is the sequence reference genome, and where these binders go, where they extended, you can run in parallel using the softwares. You can go ahead with that. So multiplexing helps in fast high throughput strategy, cost effective method, and also simplified analysis through barcode and other softwares, reducing time and other. Basically, you are moving from classical Sanger to next generation sequencing, where high throughput data can be analyzed into that. You just look into the Illumina comparisons. Illumina has evolved, Illumina next generation sequencing. They started with next 500 model, HiSeq 4000, NovaSeq 5000, and presently some other model here. You see that the model changes with the machine, the efficiency of the machine changes. For example, output range here, 20 to 120 gigabases per day, or whatever time you can see here. It goes into 6,000 gigabases from the sequencer machine. Basically, you are taking the warehouse of Sanger sequencing done in 2000 for human genome, making into whole warehouse of factories, taking them away, making into one single machine, and you are sequencing different highly competent, high amount of DNA given per hour per day. You see that? How the max read length, reads per run, you can do 3 samples per run you can do, and the cost varies with that. You go through with this, understand that. Next few slides will try to give an overview of evolution of sequencing generation, first generation, second generation, third and fourth generation, adapted from Jonathan Eisen, UC Davis. You just simply go through the evolutionary